All right, let's go ahead and get started. Thank you everyone so much for joining our workshop today where we'll be discussing how to optimize your IP workflow. I'm Afrida Rahman-Enyart. I am a scientific liaison and product manager here at Proteintech. I will also be joined by my colleague, Dr. Callie Anderson, during the Q&A session. So hopefully we'll be able to answer any questions you guys have after the workshop. Before I begin the workshop, I just want to share a few company slides with you so I can introduce Proteintech. So Proteintech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies that encompass over 13,000 targets. We also offer ELISA kits, IHC kits, antibody labeling kits, and magnetic cell separation kits. Within our Proteintech brand, we also have our humankind branded cytokines and growth factors. And all our proteins are expressed in human HEC293 cells. And because they're expressed in human cells, you know that they have native folding, post-translational modifications, and glycosylation. And then finally, we also have our Chromotech branded nanobody-based reagents. And nanobodies are essentially really tiny antibodies, and so they're ideal for the immunoprecipitation of tagged proteins, which I'll talk a little bit about today. They're also great for super resolution microscopy and protein purification. Apologies. There you go. So, as a company, we measure our success by our company's scientific success, specifically by helping them publish impactful research faster. So, this graph here on the left is showing that over time, our company's history of citations. And you can see that more and more of our products are cited each year. And as of today, we have over 200,000 publication citations. We are also one of the top 10 cited antibody companies, and we are also the fastest growing company in product citations. We also value that we are a company for scientists by scientists. Our award-winning products are self-manufactured, and we offer the largest in-house portfolio of antibodies for the human proteome. We also know the importance of product validation, and we use knockout, knockdown models for validation whenever possible. And as antibody experts, we have very in-depth knowledge of immunoassay applications and offer outstanding technical support. Um, So with that, I would like to go ahead and begin the workshop for today. So this is the agenda. So we're going to cover quite a bit of material. So I want to begin by talking a little bit about, what is immunoprecipitation and when would someone want to use this application? How to go about selecting the right antibody and matrix for your experiment, when you should use a nanobody versus using a traditional antibody for IP, and kind of the benefits that nanobodies bring to generating clean immunoprecipitations. We'll then go step by step through the IP workflow and talk about how you can optimize each step. And then we'll talk a little bit about not only what controls are recommended, but why they're so important. And that'll really come in when we covered some detailed troubleshooting. So we'll have some examples and some common issues that people face when doing IPs. And then we'll still have some time at the end for Q&A, where I'll be joined by Callie, and we'll hopefully be able to answer all your questions. So let's go ahead and begin with generally what is immunoprecipitation. So immunoprecipitation is a technique which is going to allow you to obtain a sample that's enriched with your protein of interest. So you generally are going to start out with a heterogeneous cell lysate that's going to have many different proteins in it. And then what you do is you introduce an antibody matrix that recognizes your protein of interest, which we show here in green. And once the antibody binds to your protein of interest, you can then wash away any of the unbound proteins and lipids. So then what you're essentially left with is an enriched sample that includes your protein of interest and any of its interaction partners. So why would someone want to do an immunoprecipitation? So there are several different questions that an IP can really answer. So for example, because you are pulling down your protein of interest, as well as anything else that is bound to it, it's a great way to identify any interaction partners or protein complexes. This is also known as a co-IP. So you can essentially perform co-IPs and Western blots in order to analyze things like post-translational modifications. And lastly, some people use IPs as a way to concentrate or enrich their protein of interest from a lysate. So for example, if you have a low abundant protein and you need to have a more concentrated sample. So here, right, what we're showing is very simply what an IP workflow would look like. So we'll go through each of these steps and we'll discuss ways to optimize each later on in the workshop. So very briefly, your first step is going to be the preparation of your cell lysate. So this is going to be where you take your cells or your tissue and you break down the cell membranes and essentially release all the proteins and cellular components into your lysis buffer. And then you can spin this down and all the cell components will come down. And what you're left with is going to be your lysate that contains all your cellular proteins. And that's going to include, hopefully, your protein of interest as well. And then you have your binding step. So for your binding step, that's where your antibody and bead matrix is going to recognize your protein of interest. And then your wash step to remove any of the unbound molecules. And then you take your sample and you have to elute it. And so by elution, what happens is that your protein of interest then is no longer bound to the antibody. And oftentimes, the next step after the elution step is going to be some form of analysis. So most people do Western blots. Some people do also mass spec. But today, the examples that I'll really be focusing on are doing Western blot downstream of IP. So let's move on to how to select the right antibody and matrix for your experiment. So when you're selecting an antibody for IP, there are several do's and don'ts to consider. So you want to make sure that the antibody that you select has been thoroughly validated. So make sure that you look at the validation data that the manufacturer has posted on their website and make sure that the data is clean and of high quality. The manufacturer is also going to list, what type of applications the antibody is suitable for. So you want to select an antibody that has already been tested for IP. If IP isn't listed as one of the applications, that doesn't necessarily mean that the manufacturer hasn't tested it or that it doesn't work for IP, but that they might not have tested it yet. But nonetheless, to save the time and kind of effort and the hassle of optimizing, you just want to make sure that you find an antibody that has been validated. Another important consideration is going to be to select an antibody that has a high affinity for your target. So IP is not a gentle process whatsoever because there's a lot of washes, there's detergent. So you want to make sure that your antibody stays bound to your protein of interest and that you end up with a truly enriched sample. So affinity is generally measured by an equilibrium dissociation constant, which the suppliers should most likely know. So you also want to make sure, in addition to the high affinity, that the antibody is purified. So not surprisingly, a non-purified antibody is going to be dirtier. So you might get some non-specific binding and your pull down essentially won't be as clean. So you want to make sure that you use a purified antibody. So in terms of clonality, so should you use a monoclonal or polyclonal antibody when performing an IP? People really have their preference on this, but it really goes down to your experiment and what is important. So of course, you want to select an antibody that is specific for your protein of interest, or if you have, a tag on your protein, is something that's specific for the tag. A monoclonal antibody is going to tend to have, a higher specificity to a single epitope on your target antigen, which makes it ideal for IP and also results in a lower background. Monoclonals also have a higher batch-to-batch consistency, which makes reproducibility of your experiments much easier. Um, Polyclonals, on the other hand, they tend to have a high binding affinity because these antibodies are produced by multiple B cell clones. And so this leads to a mixture of antibodies that have varying affinities for different epitopes on your target antigen. And so this can result in, improved binding under certain conditions. Since polyclonals recognize different epitopes, you also tend to have a higher yield when using them for IP compared to monoclonals. And so this can be really advantageous if you have, for example, like a low abundant target, or if you're trying to detect different isoforms or post-translational modifications. And then lastly, you want to consider the matrix, so the bead that will bind to your antibody. So porous or agarose beads tend to give lower background, and they're ideal if your protein your protein or your complex is less than 200 kilodaltons. And solid beads are going to be ideal if your protein of interest or your complex is greater than 200 kilodaltons. And the other thing that you want to consider is, whether or not you want to separate by centrifugation or by magnet. And oftentimes this is kind of a personal preference, but some people, they like the magnetic version better because it has been shown to have lower background. So after you have chosen your antibody, you can then select, how you want to immobilize the antibody on the matrix. And so the easiest way is to just use protein A and G beads. So this is the most common way that people immobilize their antibody. So basically, you incubate the beads with your antibody and you get a non-covalent interaction. If you want to have a tighter interaction between the antibody and the beads, you can also cross-link the antibody. So this is where you have an irreversible cross-linking to the protein A and G beads, and the antibody bead complex can then essentially be reused for other IPs. Or there's a third option where you can also covalently bind the antibody to the beads without the use of protein A and G. So the cross-linking and the covalent versions are more popular if you're doing IP under, fairly harsh conditions. So for example, if you have a harsh wash buffer and you want to make sure that, your antibody does not get released from the beads under these harsh conditions. And then one thing to consider, however, is that when you do have a covalent interaction between your antibody and the bead, this could essentially change the paratope of your antibody, and it might no longer bind to the epitope anymore. And so that's why some researchers opt for using this non-covalent interaction with the protein A or G beads. So this would kind of be my recommendation to kind of start with the protein A and G beads and seeing whether that works before you kind of move on to these other options. So then next we'll talk a little bit about the benefits of using nanobodies if you have a tagged protein versus using a traditional antibody. So nanobodies are really great for generating very clean IPs. So when we think about a traditional IP, we picture something like this here on the left. So you have your heavy and your light chains. However, alpacas and other cameloids, in addition to this traditional antibody, have a heavy chain only antibody. And on this heavy chain only antibody, there is a binding domain, which we show here in green. And this is what we call a nanobody. And a nanobody is quite small. It's only about 15 kilodaltons. So that's about 10 times smaller than a traditional antibody. And you can essentially take this nanobody and you can conjugate it to a bead for immunoprecipitation. So here what we're showing is an example of our GFP trap, which consists of a nanobody that recognizes GFP and that is conjugated to a bead. So for majority of my examples today, I will be using the GFP trap as an example, and we'll be looking at a protein of interest that is tagged with GFP. So here what we're looking at is a comparison between using our GFP trap for IP, so on the left-hand side here, compared to using a conventional GFP antibody for IP, which we're showing here on the right. So one of the main benefits of using a GFP trap is that it's already prepared and ready to use. So it comes pre-conjugated to the bead. Whereas with a traditional antibody, you need to prepare this and incubate your antibody with your protein A and G beads. So the GFP trap essentially saves some time there. Now, if we take a look at the SDS page and the blots here, so we have three lanes. So we have our input, which is basically going to be your total cell lysate, your flow through, which is going to be all your unbound proteins from your cell lysate after you've conducted your IP, and then your bound fraction, which is going to be your precipitated protein of interest that's bound to the beads. So you can see that when you're using the GFP trap, you have one really nice clean band in the bound section, and that corresponds to your protein of interest. However, with the conventional IP, you have, you see the correct band that corresponds to your protein, but you also have these two other bands right here at the top and the bottom, which correspond to your heavy and light chains of your antibody. And these bands, if you take a look, are actually darker than your band for your protein of interest. And so this can get really tricky if your protein of interest is especially similar to the size of your antibody chains, because then you might be playing this guessing game as to, which band actually corresponds to my protein. However, because nanobodies are so small, they actually don't appear on the gel. So remember, I mentioned that they're only about 15 kilodaltons. So they usually don't come up on the gel. Also, because the beads are covalently bound to the nanobody remains bound to the beads even after you elute with a SDS sample buffer. And so therefore, what you see on your SDS page is one single band which corresponds to your protein of interest. Also, nanobodies have a really high affinity to their target, and this leads to a very effective pull down, even when your protein of interest is expressed at low amounts. And so you can see that here in the Western blot. So you can see that when you're using the GFP trap, you have essentially no protein of interest left in the flow through, and it's all enriched here in this bound section. But if you take a look at when you're using a traditional antibody, you do have enrichment in this bound section, but you see that some of your protein of interest is still left in the flow through. So when should you use a nanobody versus using a traditional antibody? So nanobodies, right now, they recognize tagged proteins. So if you have a protein that is tagged, nanobodies might be the best option for you. So we recommend using a nano trap to do your immunoprecipitation and then using an antibody to do your Western blot detection. However, if you're looking at an endogenous protein or a protein that's untagged, you want to make sure that you use one antibody for IP and then use a different one for your Western blot detection. So for example, if you're using a mouse monoclonal for your IP, use a rabbit polyclonal for your Western blot detection. So for the next part, what we'll do is we'll go through each of the steps of IP and discuss some of the tips and tricks that we at Proteintech have in order to optimize your workflow. So the steps that we'll be covering are the preparation of your cell lysate, the binding of your protein of interest, the washing of any of the unbound molecules, the elution step, and then we'll also go through a little bit about the analysis and if you're doing Western blotting afterwards. So let's begin with the first step, which is going to be the preparation of your cell lysate. So the goal here is that you have your cells that express your protein of interest, and then you break down the cellular membranes and the cellular components, and then your protein of interest and all the other proteins are released into solution. And there are different methods and buffers that are optimized for different cell types and proteins. So this is something that is really important to kind of customize based on what protein you are looking at. So this might be some trial and error, but the preparation of your cell lysate is one of the most, if not the most important step of your IP. Because you want to make sure that your protein of interest is there, it's soluble, and that you're able to IP it. And so there are different methods and buffers, as I mentioned, but it's generally recommended that you start with a mild buffer and then add some detergent to make sure, that their protein is heavy in the solution. And depending on what cell line you use, the cell material can also differ a little bit. So we recommend that per IP to use 500 micrograms of total protein or 10 to the 6th to 10 to the 7th mammalian cells. When using other materials such as plants, because the protein expression in plants can be very low, you need to use a lot more material, so more in the gram level, so about 10 grams for plant material. Also, you want to make sure that you use fresh cell lysate whenever possible and avoid freezing. And I know that's not always possible, but it does result in much cleaner data because freezing can cause changes in protein structure and it can cause aggregation. And so that can result in getting a higher background. So after preparing your cell lysate and spinning down and removing all the cell debris, you are ready to add your antibody bead mixture in. So your antibody is going to bind to your protein of interest, and the other proteins hopefully will remain in the mixture but unbound. So here are some do's and don'ts that we have. So we recommend that you incubate your lysate with the antibody bead mixture for 1 hour and make sure that you keep the beads in suspension by slow rotation. This is really important to minimize background. If you incubate for longer or you don't have the beads in suspension, you will see that higher background. And additionally, if you have a protein that is expressed very low, then it might not bind. It might not have the ability to bind to it. So you want to make sure that you avoid any of the settling of the beads, but also you want to make sure that you avoid too much mechanical stress. So there's like that happy medium. So the slow rotation is going to be the ideal way to do this. Um, in terms of volume, so we recommend that you have your volume for your IP between 200 to 500 microliters if you're working with a 1.5 mil Eppendorf tube. And that way you can ensure that there's enough room to mix properly. We also recommend that you incubate your IP at 4 degrees and in a mild buffer in order to keep, everything happy and in order to avoid proteins from unfolding. If you have too much salt or too much detergent, you kind of risk this aggregation and some of your proteins unfolding. So the next step is going to be your washing step, where you remove any of the unbound cell lysate components, and your protein of interest is going to remain bound to your antibody and the beads. So when incubating the IP, you want to make sure that you use a mild buffer. But when you're doing your wash step, you want to add a little bit of a harsher buffer. So up kind of the detergent a little bit so that you can make sure that any of the proteins or cellular components that are binding to your antibody or beads unspecifically can get washed away. Oftentimes I say it's very similar to like washing your hands, and the other proteins are like dirt. And so you would use more soap if you have more dirt. So you want to just make sure that you kind of up the detergent in your lysis buffer. It's also recommended that you wash for a couple minutes, 3 to 5 times with about 1 to 1.5 mils of the buffer. If you wash too long or too many times or with a very large volume, you not only risk, washing away the unspecific binding, but also your protein of interest. So you just want to wash away the unspecific binding. So for the last step before you load your sample is going to be your elution step. In this step, the protein of interest is released from the beads. And so there are a few different options for how you can elute. The most common option is the denaturing elution with SDS sample buffer. And during this process, the antibody and the protein of interest are both denatured when they are heated, and the protein of interest is then released. Another option is doing an acidic elution. So for example, using something like 200 millimolars of glycine solution. During the process, the interaction between the antibody and the protein of interest is going to be disrupted. And depending on the protein, it might stay in the native state or it might denature. So this is a good option if you're trying to look at proteins that are in their native state. The last type is proteolytic elution by using, for example, TEV protease. So here you would need a cleavage site between your protein of interest and your tag when you're cloning. So basically, your antibody would recognize the protein of interest and you would cleave the tag, so your protein of interest would then be released in the solution. Um, so after you've completed your elution step, you can then do some downstream analysis to analyze your IP. So for example, like Western blot or mass spectrometry. Sorry, I cannot talk today. Mass spec. So here, I just want to show you how your analysis might look like on SDS page and Western blot. So on the left side, we have your SDS page, and on the right is going to be your Western blot. So what we're showing here in this example, we're going to be using, as I mentioned, the GFP trap. So if your protein of interest is a GFP expressing protein, so we're going to assume that we are IPing for the GFP trap and then Western blotting using an anti-GFP antibody. So the first lane you're going to analyze is your input. And so this is going to be your total cell lysate. So on your SDS page, you're going to see many, many different bands. And when you probe this lane on your Western blot, with your antibody that corresponds to your protein of interest, you're going to see hopefully the band that corresponds to your protein of interest. The next lane that you're going to have is going to be your flow through lane. And this is going to be anything that's left over in your cell lysate after you've done the IP. So this should be all your unbound proteins. So when you look at it on the SDS page, it's not going to look much different than your input. But when you look at your Western blot, you should essentially not see a band if your IP worked, if you're blotting with your anti-GFP antibody. And then finally, you should analyze your bound fraction. So the precipitated protein that has bound to the beads. So if your protein is of high abundance, you're going to see a single band on your SDS page. I wouldn't worry if you don't see this band on your SDS page because, as I mentioned, you need to have a pretty high expressing protein in order to see this band. So sometimes it doesn't appear. But on your Western blot, you should be able to see a enriched and strong band that correlates to your protein of interest. And this band should be more strong than what you observe in the input. So these are kind of the normal analysis that you would do when doing an IP, but controls are also going to be really important. And so that's kind of what the next couple sections are going to show you. So let's discuss what types of controls you should run when doing your immunoprecipitation. So one very typical control is going to be your positive control. So this will be, this is going to give you a good idea of, is your IP setup working? Are your buffers good? So the idea here is that you would see the enrichment of your protein of interest. So if you're working with a protein with a tag, then you basically can have a tag-only condition. If you are working with an endogenous target, it can be a little bit more difficult, but you can use, for example, like a purified recombinant protein. So with a positive control, you would expect to see your protein of interest in the input, as well as enriched in your bound section. And if you see this, then you know that your IP setup is working and that you're using all the right buffers, you're incubating in the right temperatures, et cetera. More importantly than your positive control is going to be your negative controls. So the main goal here is to make sure that you don't have any unspecific binding when your protein of interest or your tag aren't present. And so this is going to be really important when you're, looking at your data and trying to make a conclusion out of it. So when you're working with tag proteins, we recommend using a cell lysate that contains the protein of interest without the tag, and then working with endogenous proteins, you want to ideally use, a genetic knockdown or knockout if that's available. And so here in our example blot, you can see that a negative control with no GFP would show no bands when you're using a GFP antibody. Another important negative control is to make sure that you have no unspecific binding to the bead matrix. So sometimes proteins can unspecifically bind to the beads themselves. So based off of whether you are using, an antibody or antibody, you can use blocked beads or the protein A or and G beads without the antibody to see, what are your beads binding to in the cell lysate. You can also use an isotype control antibody, which is going to be the same isotype as the antibody that you are using for IP. And you can incubate the control antibody with the protein A and G beads and see, what that binds to. So then when you look at, your Western blot, you should be able to see your protein of interest in the input and in the flow through, because remember, you're not using your GFP antibody that for your IP. This is going to be your negative control, and essentially nothing in your bound section. So this is going to basically tell you that, you have a very clean IP and nothing is binding unspecifically to your bead matrix. So for the last section, I want to go through some examples, some common troubleshooting issues that people have when conducting IP, and kind of how to tackle these issues. So in the examples, again, similar to all the other examples that I've shown you today, we're going to be using a GFP tagged protein of interest that is immunoprecipitated with GFP trap and Western blotted with an anti-GFP antibody. So we're going to take a look at 4 common issues that researchers deal with when performing IP. So we'll go through each of these issues. So for the first issue, that's having high background in the bound fraction. So sometimes you might see additional bands in the SDS page for your bound fraction. So ideally you would only see the one band that corresponds to your protein of interest, but sometimes you see multiple bands. And this unfortunately can also show up on your Western blot. And one of the reasons for this type of high background could be due to the high concentration of cell lysate. So for example, if you have too many cells or not, you haven't diluted your samples enough. The other issue might be that the cellular proteins or the protein of interest is degraded or aggregated. If your protein of interest is degraded, you might see, a lot of smaller bands below your protein of interest. So that would be a good idea as to whether or not your protein has degraded or not. So what can you do to resolve this issue? So as you will see with many of the troubleshooting issues that we're going to discuss today, uh, it's going to start with optimizing your cell lysis. So making sure that you test different methods. You also want to try and find the right amount of cell material per IP. So for the binding step, when you have high background, it's generally beneficial to shorten the binding step. So we recommend doing, this step at 4 degrees for a max of 1 hour. So the longer you incubate, usually what will happen is only the background increases. You can also pre-incubate your protein A and G beads with your cell lysate in order to reduce anything that is unspecifically binding to the beads. And so this is also known as pre-clearing. If your tissue is if your cells are undergoing protein degradation, you can also add protease inhibitors. So again, if you see a lot of smaller bands below your protein of interest, this might mean that your protein is being degraded and your sample might benefit from using protease inhibitors. In terms of washing, when you see high background, you can use a harsher buffer. So try to increase the salt or the detergent concentration to get rid of any of that unspecific binding. And finally, sometimes something as simple as, just using a new tube for your last wash step might help with the background. So oftentimes, we blame our antibodies or buffers and other reagents, but sometimes it can just be the fact that proteins love to stick to plastic, and so might just need a new tube. The second case, which is a really, really disheartening one, is if you don't see your protein of interest in the bound fraction and it's all in the flow through. There could be different reasons for this. So, for example, the protein of interest might be insoluble, or it could be unfolded and then aggregated or degraded. There could also be insufficient mixing. So, the antibody didn't have the opportunity to bind to the protein of interest. And it's also possible that there just wasn't enough bead slurry applied to your sample. So you essentially, you maximize what your antibody can bind to, but it isn't sufficient to see a band. So oftentimes in IPs, your antibody is your limiting step. So you kind of want to make sure that you find that happy medium as to how much antibody and bead slurry you need to add to kind of maximize the amount of protein that you can see in your IP. And then in more rare cases, it could be possible that your protein of interest is too large to enter the matrix pores. So how can you resolve this issue? So if your concern is your protein aggregation or degradation, then you might need to test different protein expression and lysis methods again. So this troubleshooting is obviously going to occur before you even perform the IP. And this type of degradation usually occurs during the preparation of the lysate. So I know I've mentioned it a couple times during this workshop, but the lysate preparation is really going to be key to having an optimal IP. If the issue is occurring during your binding step, then want to make sure that you're mixing properly. So mix by end over end rotation so that everything is mixing. You want to also make sure that you're using 25 microliters of slurry for one IP reaction and testing different bead matrices. So trying porous versus solid beads and seeing what works better in your experiment. So then the third issue that can happen is if you see no protein of interest in both the input and the bound fraction. So one possible explanation is that your protein of interest is just too low. The other reason might be that the protein of interest is there, but it is insoluble. So basically, it gets stuck in the cell debris pellet. So some solutions for this is going to be to test different protein expression and cell lysis methods, or to try and use, more cells or a higher concentration of cell lysate for your IP. It's also possible that your Western blot antibody isn't working well, in which case you might want to try, having a positive control and also testing different antibodies and seeing what works best. And so now for the last case in this chunk, when you see low amount of your protein of interest after eluting with SDS sample buffer. So you essentially are going to have less protein in your bound fraction than in your input. So not what you would expect if your IP was successful. So in this example, you can see that, there's still no protein of interest in the flow through. So the binding is clearly working, but the reason is because, you have insufficient elution, and that is resulting in no enrichment of your protein of interest. And this actually happens more often than you would think. And the solution here would be to use at least 50 microliters of SDS sample buffer in order to ensure that everything is getting mixed properly, and that the SDS sample buffer and the beads are mixing. You also want to make sure that you mix the beads before and after boiling, and you want to make sure that you boil the samples for 5 minutes at 98 degrees. I also want to touch a little bit about co-IP and troubleshooting for co-IP because it is quite popular. So co-immunoprecipitations are used in order to identify interaction partners of a particular protein. So for example, here we're using a GFP trap, similar to all our other examples, in order to pull down a GFP expressing protein. And this is going to be known as our bait protein. If other proteins are bound to our bait protein, these are going to be known as our prey proteins, and they will get indirectly precipitated by the GFP bait. So therefore, if you essentially do an IP followed by a Western blot or mass spec, we would be able to not only analyze the bait protein, but also the bound prey proteins as well. So in terms of what type of controls you should run for a co-IP, so you want to make sure that you have a positive control. And so using, a GFP as an example, you want a sample that you know expresses this protein, but doesn't express your prey protein. You want to make sure that you also include a negative control, and this is going to be your IP that doesn't have your GFP bait in it. And this is going to tell you whether your prey protein is being precipitated unspecifically or, by the beads. And then finally, you want your experimental condition. So basically having a sample that expresses both your bait and the prey protein and IP'd for your bait protein. Another good idea is to do reciprocal IPs. So for example, after doing an IP, the IP here, you would want to switch so that your prey becomes the bait, and then seeing whether or not when you IP for the prey, does the bait also come down with it? So here are some common issues that might arise when doing a co-IP. So for example, for these examples, again, we're going to continue using this GFP trap and where we IP for the bait protein and we're looking at the prey protein. So we'll be Western blotting for anti-GFP and anti-prey antibodies. So for the first issue, if you see essentially no pull down of the GFP bait in the positive control or in your experimental experiment. So since the GFP bait is not pulled down, your prey protein obviously is also not going to pull down. However, if you look at your positive control where you just looked at GFP, so it's not GFP attached to your bait, you do see that it gets pulled down. So here, obviously, it's showing that the pull down is working. So then the issue is because of your GFP bait. So it's possible that the GFP bait itself isn't soluble or it's unfolded or there's only a small amount that is precipitated. So the solution here would be to optimize the conditions for your GFP bait first. So focus on the bait and then focus on the prey. So we discussed this earlier in the case where we saw no pull down of your GFP protein of interest, but similar to that, some of the methods for troubleshooting are going to be, again, to optimize your protein expression in your cell lysis, and then optimizing your IP in general. So making sure everything's properly mixed, testing different matrices, making sure that you're using 25 microliters of the bead slurry per IP. The second case is if the IP of your prey doesn't work. And this is probably the most common case that our tech support sees. So we essentially here see there's no pull down of the prey, but the positive control looks good. See both the GFP bait and the GFP are being pulled down. But in the experimental condition, the issue is very clearly because of the prey. So some of the concerns might be the prey might not be soluble, or there's only a small amount that is precipitated. And so the solution here, if you're expecting to see the prey come down, is that you want to test different cell lysis buffers and binding buffers. So in this situation, optimize the IP for the prey. And then also consider whether there really is an interaction between the bait and the prey protein. So this is where a reciprocal IP is going to really be beneficial because you can IP for the prey and see if the bait comes down with it. So that essentially gives you two different experimental conditions to look at. It's also possible that the tag of the bait interferes with the interaction. So you might need to place the tag in elsewhere on your protein. The third case, which is also fairly common, is that your prey protein is being pulled down unspecifically. So you can see it basically in the negative controls. And although you don't see your GFP bait, your prey is still being pulled down. So again, this is going to be an issue with the prey. So some of the reasons for this is that, your prey again might be unfolded or aggregated so that it's binding to the beads unspecifically. So the solution here would be, again, optimizing conditions for the prey. So focus on the prey protein first and then focus on the GFP bait. So you want to make sure that, just when you're making it, when you're testing for the prey protein, do the test without the GFP bait and see if you can just optimize the IP for the prey. And once those conditions look good, then test with the GFP bait. So basically, through all the troubleshooting examples that I showed you, you can see just how important controls really are. So overall, these are the take-home messages that I hope you guys got from the workshop. So first, we talked a little bit about making sure that you choose the appropriate nanobody or antibody for your IP, making sure that you look through the validation data and that not every antibody or nanobody is going to be suitable for IP. So make sure that you find something that is validated. We also discussed a little bit about, the sample preparation. So especially during the troubleshooting section, sample preparation is always key, figuring out the correct lysis buffer for your protein. Unfortunately, there's not like a magic recipe. It's really protein dependent. So kind of optimizing those conditions. We also went through each of the steps of IP and talked about the fact that each of these steps can be optimized so that you can have the best results and, kind of optimize your entire IP workflow. And then we talked a lot about controls and the importance of controls, not only for getting meaningful results where you can kind of conclude from your data, but also for helping you with troubleshooting and making sure that, your IP is optimized to its fullest extent. So before we begin the Q&A, I just want to talk about the fact that we do have other videos. If you liked this workshop and want to learn more about other applications, we do have a video hub on our website. We also post about our upcoming events. So we have other workshops. So many of you might know that this is immunoassays week for us. So each day we've been focusing on a different immunoassay. Tomorrow we're going to be focusing on Western blot. So we do have an events page where we talk about any of our upcoming events and webinars. And then as an attendee of today's workshop, you will receive a follow-up email that will have a link where you can request a free antibody or nanotrap sample. You will also receive a certificate of attendance as well as a recording of this webinar. So with that, I'd like to thank everyone for joining the workshop. I'll now be joined by my colleague, Dr. Callie Anderson, who will help out with the Q&A. So thanks, Callie.
Hi, Frida. Thank you. It was a really fantastic presentation. I definitely learned a few things from you there about IP too. We've had a number of questions in the chat, so I've got through some of the answers to these, but we had some interesting, more general ones, which I think that we could go through just now. So one thing I see come up in tech support quite often, and I think it's a nice thing to point out, the question from Diego, which is asking, they see a band in their IP fraction, but not in the input. And they're wondering if they should change the lysis method, or maybe it's that the antibody they're using is not sensitive enough for the Western blot.
Yeah, so that's a really good question. I would definitely test out different antibodies. It might be that, if you're using a polyclonal antibody, maybe try a monoclonal antibody, something that's a little bit more cleaner. You might also try out your lysis method, but it might be easier to just start off first testing out different antibodies before you kind of get into changing up your whole lysate preparation. Callie, I don't know if you had anything to add to that.
Yeah, definitely. I agree. And I also think if you're getting a nice band in your bound fraction. It might just be that the antibody you're using is not sensitive enough with the amount of protein you're loading. If it's really low abundant in your lysate, it might be one of these cases where you really need to enrich it to get the band in the IP coming up there. Okay, we had a question about a recommended lysis buffer to start with, because I know that we talk about how important the lysis buffer is. So I did flag in the Q&A a link to one of our protocols, which really gives you our recommended lysis buffer for mammalian cell lines. So it's a really good starting point. And obviously, depending on your protein of interest or your sample type, you can adjust this. And looking at the literature is often a really great place to start, particularly anyone who's used the same sample type for the same protein. It's always best to follow what someone else has seen to work beforehand, definitely.
Yeah, absolutely. And like just considering cellular localization too, if you're looking at something that's more of a membrane protein that doesn't necessarily need all the harsh detergents, keep that out of your lysis buffer. You want to make sure that you kind of optimize your lysis buffers based off of where your proteins are cellularly expressed, because you want to kind of manage how much of your protein is being unfolded or degrading or anything like that. So yeah, as Callie mentioned, we do have a lot of great resources in terms of recipes for lysis buffers, especially when looking at different proteins.
Yeah, definitely. Okay, I had another question which I thought would be good to go over. And this was quite simple, but it was which is the best dilution method? So I did write out an answer here, because it really depends on your downstream application. So we recommend several different dilution methods, and they have different efficiencies. So using your 2x SDS sample buffer is a really efficient a method to elute your proteins, but obviously these will become denatured and you're then more limited, maybe in your downstream applications, for what you can use that sample for. Acidic elution can be really good, allow you to elute the native proteins. You do need to test this because many proteins will be fine after you perform the neutralization step, but there are some which might be impacted. So if you need them to be active, it might not always work, but it's a really good option. And I know Afrida mentioned about cloning the TV cleavage site in, and I get many customers who I suggest this, and they wish they had known that this is a possibility upstream, because obviously that comes back to going back to cloning and getting that in there. I'm not sure if you mentioned actually during this presentation, if you are using something like a peptide tag, so something like V5 or a spot tag, you can also use a competitive peptide dilution. And that's a really efficient method to elute. It's not possible for GFP, which has been the example we've used here, or RFP, but if you are using a peptide tag, that's a really nice way to elute from the trap as well.
Yeah, absolutely agree with everything you said. That was a really good summary. And the other thing to consider is, again, what are you trying to look at? So in terms of acidic elution, acidic elution is going to be really ideal if you need to maintain your protein structure and making sure, because sometimes those proteins don't get denatured. So it's better in terms of not kind of affecting those non-covalent bonds. And so the other thing is that, if you're looking at large protein complexes, acidic elution is also something that might work better than SDS sample buffer. But generally, SDS sample buffer is kind of everyone's go-to to start with. But yeah, but I think, Callie, you summarized it really well.
Yeah, I think it's also worth mentioning there's you can also do some on-bead downstream assays, so on-bead digest for mass spec. So you don't even need to elute the samples, or it's also possible to do some enzymatic assays with your proteins on bead. So there's lots of different options with using the traps, definitely anyway, because they're pretty robust. I have one more question to do, and this was just suggestions for non-specific binding, which is resulting in the isotype control. So my guess here is this has nothing really to do with your antibody that you are using the IP, but maybe more to do with the beads that you're using. So proteins in the lysate, which are just sticking non-specifically to the beads. So I could definitely suggest altering perhaps the sodium chloride or salt concentration in your buffer, or the detergent concentration. Anything that's going to increase the ionic strength of the buffer might help to get rid of those non-specific interactions. Of course, you've also got to balance that with things that are going to interrupt your antibody or nanobody binding to your protein of interest too. Did you have any
Yeah,
other suggestions, Afrida?
yeah, I was gonna just add to that. The other thing that you can consider doing is pre-clearing your cell lysate. So just incubating with your beads, and then I know that this can be a little wasteful of beads because then you have to switch out your beads with new beads. But it's a really good way of kind of getting anything that's unspecifically binding to your beads to get that out of your cell lysate. That's a hit or miss though, because then you're taking away some of the proteins from your cell lysate. So is it truly like, a biologically relevant sample anymore? Because you're like removing those proteins. But this is a good way if you're kind of getting a lot of background.
Yeah, absolutely. I think if you are using our nanotraps for this, a really nice way to pre-clear those is to actually use an unrelated trap. So if you're using GFP trap, do that pre-clearing with the RFP trap, because you not only have the beads there, but you also have another nanobody there. So if it's a case that this protein just binding non-specifically to a nanobody as well. So that's a nice way to do it. Great. Well, we have some more questions, and I think if we've not managed to get around to answering your questions now, we can definitely get in touch with you. And feel free to email either Frida or myself or our events email address with any follow-up questions that you have. I know Frida mentioned it, but we will send out the slides, PDF copy of the slides, but also a link to the recording of the webinar for you to come back to too.
Yeah, absolutely. And I just want to thank everyone again for joining, and again for the wonderful questions. I'm always happy and amazed with how many really good questions that we have. And yeah, as Callie mentioned, we'd be happy to reach out and answer any of the questions that we didn't get to live. And if Callie, I don't know if you mentioned, but I want to mention this again, that tomorrow we do have our Western blot workshop. So make sure, if you guys haven't signed up for that yet, to sign up for that. That will be given by you and Amy, right, Callie?
Yeah, Amy will be presenting, but I will be hosting again. So I'll be online to help with questions too. So hopefully we'll see some more of you online there too.
Yeah, awesome. All right, well, thank you. So with that, I'll go ahead and end the webinar. I hope everyone has a great rest of your day.